Hi, everyone. I'm so happy to be here with you today. I have seen so very cool presentations. And rather to be a talk with computational method, development of computational method, I would like to use this talk to talk about a wide range of computational methods applied to cancer research. So I'm very interested. So my project is based on a very particular tumor called Medulloblastoma. I think we have a presentation of also about this tumor. So Medulloblastoma is quite interesting because it's one of the most aggressive pediatric brain tumors in children, and it usually arises in the cerebellum and is typically common in even babies. So that's something that we need to think about when we are trying to understand this type of cancer. Now, the thing about medulloblastoma is that it's quite heterogeneous. So over the years, there has been many robust molecular profiling that have enables to identify that medulloblastoma is composed of different molecular subgroups. And over the years, when you have more robust analysis, you can even identify subgroups within the subgroups. So far it has been identified 12 subgroups, but we can split it into four main groups. And this, for the example of from these groups, the most aggressive is called group 3, which is known to be almost metastatic most of the cases. Like 50% of the patients with group 3, they will already present metastases at moment of diagnosis. The problem with metastases in this type of context of this brain tumor is that it's usually spread like you spread like butter in bread. So you cannot resect that, you cannot operate that. So that's why it's very hard to get samples to understand the metastatic compartment instead of the primary. So for that, to try to solve this problem, we try to generate a metastatic model of this tumor. So what we do is that the primary tumor is quite easy to get, actually. So this is just an injection that we get from cells from patients, and then we inject it into mice orthotopically. That means in the site of the location of the tumor, in this case in the cerebellum, and then we wait for the mouse to develop clinical signs, and then we harvest the tumor. But for the metastatic, for the metastatic model, that's a little bit more challenging. So what we do is that based on in the notion that it was discovered that for some reason that we don't know, metastatic cells tend to leave the nervous system, which is quite rare in a brain tumor, and they go through they go circulate and enter bloodstream. But for some reason, they like to come back to the nervous system, and then that's why how they at some point they develop metastasis in that way. So with that notion, so we of this notion, we inject the cells in the flank, and then we wait to have this tumor. And then we resect this tumor to give a little bit more of time, and after 1 month and a half, you will have metastatic back to the brain, which is so very, very exciting and very interesting to see. So when we harvest these samples and we take it for molecular profiling, and then we have been able to have both the primary and metastatic and say, okay, which are the differences between the two? So what we have been doing is that we have trying to do a wide range of molecular analysis, transcriptome, and genomics. So today I'm going to talk about the transcriptome of metastatic compartment. So one of the challenges that we had is that, of course, our samples, when we harvest the tumor, they come with the contamination of the host, in this case of the mouse. So we have different bioinformatic tools like Xenome that help us to clean the mouse reads and have nice filtered human reads that we can analyze. So with that, we do the famous tool Salmon that's very fast to do to get all the quasi mapping and get the reads to do the transcriptome. And what we have identified is that indeed metastatic medulloblastoma is quite different from the primary, which is clinically is very relevant because it's not working the way that we treat medulloblastoma. We treat it in the current standard way. So knowing this, having this in mind, so we need to know about better strategies to try to target metastatic medulloblastoma. So what we have identified is that in the metastatic compartment, we have an enrichment of several pathways that includes related to MYC targets, unfolded protein response, and E2F targets, which tell us the notion that this could be these metastatic are happy to do a lot of enhancement of the machinery, translational machinery, do a lot of proteins, something that we have called hypertranscription. So we know that these hypertranscriptive cells are doing this progression and the metastasis. So what we are also trying to do is trying to see if we can correlate our transcriptomic data to some already single cell data sets that of the medullostoma that have been published elsewhere. So we try to do the convolution, which is a process that, having this single cell as a reference, we can estimate the cellular fractions of our transcriptome. And what we did, we use as a reference, we use this famous data set of single cell of medulloblastoma. In this paper, they identified 3 main populations. So one population that is mitotic, another population that is neuronally differentiated, and a third that is called progenitors population. So what we identify using the convolution is that in the metastatic samples seem to have an enrichment in terms of the progenitors population. And that's very interesting because the progenitors population, if you read that paper, it will match with the phenotype of the transcriptome that we were seeing earlier. So that was interesting finding. So we try to validate these results. We try to do our own data sets of single nuclei. So we try to do, I believe this is the first single nuclei data of metastatic medulloblastoma that has been ever done. So what we did is do the libraries, and we have the challenge of the mouse contamination. So it was a challenge also to clean the single nuclei data from the mouse reads. But finally we were able, aligning and taking the mouse cells out. So what we identified is that our results matched with the single nuclei reference. We identified the 3 populations, progenitors, the mitotic, and the neuronally differentiated. And the interesting thing is that we wanted to see if really we saw changes in the cell subpopulations proportions between primary and metastatic. So using a very cool tool called MILO-R package, which is a tool that explores local neighbors of cells using a KNN neighbor, and that it can help to stimulate subtle differences between conditions. So what we could see is that you see the color in blue are the leptomeningeal cells, and in yellow are the primary, and then you can see that significantly there is an enrichment of the progenitors population in the metastatic. So it is for us very interesting to know that probably this could be a good start point to start thinking, okay, maybe there's a way to target or try to prevent leptomeningeal disease. Maybe we need to make more research about these progenitor cells that have this enrichment of phenotype. So in conclusion, we could say that metastatic medulloblastoma seems to have hypertranscription, which is an outregulation of protein translational machinery synthesis based on the results that we have in the bulk. And based on the convolution and single nuclei, we really see that these are coming from this population, the progenitors one, and that seems to be probably the ones are driving aggression in medulloblastoma. So yeah, so what are the implications in the clinical setting? We are trying to see if we can validate this in vivo. That means probably treating mice with a proteasome inhibitor. That means trying to alter their status of target the unfolded protein response they are having. Like that, we can make them more vulnerable and treat them, and then maybe that could be useful in a clinical setting. So this is my lab. So we focus on pediatric cancer. We focus on, as you could see, in my case, I focus on pediatric brain tumors, but also my lab also do sarcoma. And of course, we do a lot of bioinformatic tools to try to make to solve all these biological questions that are relevant for this type of aggressive tumors. So also you can find that's my social media. And if you are interested to keep talking, you can also find me in the ISCB Student Council booth. You can find them every day outside in the lunch room. You can find all the booths, and then you will find a student council, and I will be there. So thank you so much for your time listening, and I'm happy to take any questions if you have any.
So thank you very much, Anna. So there is any question for Anna today? No? Please.
Thank you for the talk. I was just curious, have you considered like integrating spatial information like spatial transcriptomics into the study?
Yeah, that's it's an interesting question. Yeah, we would like to. Right now, it's something that we are considering, but so far we haven't done that. But yeah, that would be super cool also to do something like that. Yeah.
Thank you.
So let's thank again Anna. If there is no other question, thank you again, Anna, for the nice presentation and